हेलो फ्रेंड्स, गुड मॉर्निंग टू ऑल। सभी को मेरा नमस्कार। My name is Ashok Rupner and I am speaking from the campus of the IISER Pune. So let me show you the building of the IISER Pune. So this is the beautiful building of the IISER Pune, and at the entrance of this building there is a very nice quote. And what is that? The IISER Pune, where tomorrow's science begins today. So this quote is very appropriate for this series. That's why I am showing this quote you again and again. Now let me show you one more picture that shows the campus of the IISER Pune. So this is the beautiful campus of the IISER Pune. Let me show you again one more department. So this is the outreach department of the IISER Pune, Srimati Indrayani Balan Science Activity Center. Actually here we do lot of activity for the students, teachers. As you know, every second Sunday and the fourth Sunday we always bring something new for you, and today's topic is also very interesting. And we have invited our IISER faculty Dr Girish Ratnaparkhi for this talk. And so let me introduce the Dr Girish. Dr Girish did his BSc Zoology from the Ferguson College. Then he did his MSc Zoology from the University of the Pune. He did his PhD in molecular biophysics from Indian Institute of Science, Bangalore, and postdoctoral studies on developmental biology at NCBS, Bangalore. Then he moved to the University of the California, Los Angeles, USA. Presently he is an associate professor of genetics at the IISER Pune. Now let me invite Girish. Now Girish, it's over to you. If you have any question in your mind, please write your question in the chat, and definitely Girish would love to answer your question.
Today my goal is to introduce all of you to the world of animal development. The seminar will be in English and it will be targeted towards middle and high school students. And for those of you for whom English is not a primary language, you will find that images and the videos which I will show you during the course of this talk will really help you in understanding animal development in a clear manner. So now let me go to my first slide. So as all of you are aware, life originated on our earth almost 3.5 to 4 billion years ago, and the age of the earth currently, as we have calculated it, is around 4.5 billion years ago. So in the Precambrian era, prokaryotic like organisms arose, and they evolved into the more diverse kinds of life which you see on the earth today. Can I have the next slide, please? Next. So today on our planet Earth, which shelters us all, there is a huge diversity of life. Roughly we believe that there are anywhere between 10 to 100 million species of organisms on our planet. Notable amongst these are the number of microbes. There are about 10 raised to 28 microbes. That's a lot of microbes. That's 10 followed by 28 zeros. And amongst all the organisms, phylum Insecta is really very diverse, with approximately 10 raised to 19 insects which are seen on our planet. Now this huge diversity of life which we have on our planet comes in different shapes, sizes and morphology, and the whole science of developmental biology is trying to understand what leads to the formation of such diverse animals of different shapes, sizes and morphology. Can I have the next slide, please? So here are just some pictures which I have taken from Google Images, and these represent a very pitifully small fraction of the varied and very strange animals which exist on this earth. For example, on the left hand corner you can see the red lipped batfish, on the right hand corner you can see the very large narwhal whale, and if you go on Google and look for different animals, strange animals, you will find many such fascinating pictures on the web. Now the question for animal developmental biologists is, how do these different forms, what are the processes which lead to these different animals to form? And to make you understand this, I will go to the next slide. Now all animals on this earth are connected, we are all cousins, and the underlying connectedness is basically dictated by the fact that all of us store information in our genomes inside the nucleus of our bodies in the form of nucleic acid, either DNA or RNA. Now, we know from many years of understanding that DNA is the storage molecule, it stores information. DNA codes for RNA, and RNA finally codes for protein, with RNA and protein being the two executive molecules inside the cell. Now, what you see in front of you is the tree of life, and as you can see, vertebrates, plants, archaea on the right hand side, prokaryotes, all of us arose from a common ancestor, and all of us have a common universal genetic code. Now, the spread and variety of life on the planet today exists in spite of many extinctions which have happened in over geological time scales. Can I have the next slide, please? Now, the first major concept which I want you to understand is that all living organisms are made up of cells. There are organisms which are single cell organisms, but there are also many organisms which are multicellular, which means that they are made up of many cells. Now, each eukaryotic cell, which is a cell which is in front of you, has a nucleus, which is the pink and red spot in the middle, and it has a cytoplasm around it, and the nucleus and the cytoplasm are enclosed by a boundary of membrane which is in black. Now, the DNA resides in the nucleus, and in the nucleus it stores all the information which is required to make the cell function, and it also stores all the information which is required to develop an organism. Compared to a cell which is approximately 10 micrometer in size, there are other organisms, for example, with COVID being a serious problem in our lives currently. On the right hand corner, you see that scale for the COVID virus, which is extremely small. Can I move to the next slide, please? Now, these cells are the building blocks of all living organisms, and using Lego blocks, I have shown you a representation of how cells can make up an organism with a human being as an example. Can I have the next slide, please? Now, all development starts with a single cell, which we call as the zygote, which is shown on the left hand side. To make an organism, and I have shown an example of the ex-president of the United States, Donald Trump, on the right hand side, who, like all of us, had approximately 10⁻13 cells in our body. We start from a single cell, the zygote. Cells divide into two, and they divide again and again and again, and somehow these group of cells work around to make an organism. And I have already shown all of you the very strange diversity in terms of shape and size of the organisms which are present on this earth. The next slide, please. Now, organisms can be diverse and can be on different size scales. For example, the zebrafish, which is a marine organism, the seahorse, which is a marine organism, is on the centimeter scale. Elephants and humans are on the meter scale, and head louse, which is a bug which lives on our hair, is in the millimeter time scale. The number of cells in each one of these organisms range from a few thousands to a few millions. And what developmental biology, which is the topic for today, is all about, is understanding how a single cell which divides into so many cells can shape and form the diverse amounts of organisms which I have shown you in the previous slides. And in the biological sciences, we call this pattern formation, or we call this the developmental program. Can I have the next slide, please? Now, all of you know about life cycles and the circle of life, and what this means is, as time passes, organisms reproduce, they make young animals, these young animals grow to be adults, which in turn sexually reproduce to make more animals, and this cycle continues for vertebrates like humans, for insects like invertebrates, and also for microbes. Now, in the next slide, what you will see is a life cycle of an organism which you are well aware of, which is the common frog. A frog takes approximately 4 to 6 months to develop, starting from a single fertilized embryo or a zygote. The life cycle shows you that the embryo makes a tadpole, then the tadpole will finally grow into an adult frog, and after maturation, the adult frog, the female frog, will mate with the male frog and make eggs. In the next slide, I will show you a movie on frog development. Please pay attention to all stages of this movie. This is the single cell from which development starts, the zygote. The cell is now dividing into two, and it will keep on dividing till it makes a large number of cells, and these large number of cells will finally shape and form a frog. These are the very early stages of animal development. As you can see, cells keep on dividing, and at this moment in development, the size of the developing embryo does not change. The yellow part is the yolk, which is providing nutrition to the developing eggs as they keep on dividing. Now these cells which are stuck together will take shape and form of an animal. What you see on the top over here is the formation of the nervous system, the neural folds which will fold and form most of the nervous system of the animal, and which is shown here with a triangular arrow. Now watch carefully how these cells, as they divide and shape and form a tadpole and then form a frog. This is a very dynamic process, and cells are still dividing slowly, but a lot of attention currently is on shaping and forming these group of cells into an organism. Now this organism will hatch into a tadpole, and the tadpole will first swim around, eat a lot, and then grow legs and finally shape itself into an adult frog.
Or larvae.
So now you will see this is a tadpole, it will move around, the legs will be developed, the tadpole still has a long tail which will be degraded, and you end up with a frog. In the next slide, we will now look at another animal which is basically a chicken. Now many people say, how does a chicken come from an egg? Now that is a very developmental problem, which is to define how a single cell which is growing inside the egg, which you are very familiar with, will turn into a chicken. And what I do as a scientist is I study another organism, the next click please, which is the fruit fly Drosophila melanogaster, which also forms from a single egg, and which is something I will talk to you about near the end of this seminar. So let us first look at chicken development. The next slide please. Now chicken development follows the same pattern as it with all development. You start with a zygote on the left hand side, there is cell division, you have the 4 cell stage, the 8 cell stage. These cells form a football like blastula, which is a hollow ball with fluid inside. This blastula undergoes a series of very dynamic shape changes, which is called gastrulation. Gastrulation creates 3 germ layers, which you will read about in 7th or 8th standard, and these 3 layers of cells called the ectoderm, the endoderm and the mesoderm are finally responsible for all tissue which is there in animals. Can we go to the next slide please? Now this is a human embryo, and though I will not be talking to you in detail about human embryogenesis, the next slide will show you links, will show you pictures of a human being developing, and the slide after this will show you websites to go to where you can watch a human embryo develop and also get a lot of 3-dimensional information on the developing human embryo. In the next slide, as an example, we will look at another vertebrate which develops similar to humans, which is the chicken embryo. And what you see over here are the different stages of chicken embryo development, starting from a single cell all the way to a hatched embryo. Now the movie on the next slide will now show you how a chicken embryo develops as an example of developmental biology. So it takes 21 days for an egg to hatch, and let us see all the stages. The yellow background is the yolk, which is providing nutrition to the developing embryos. You can see the limbs are forming, the brain and the eye are formed by day 4. By day 6, the digits are forming. The beak develops at this time. Feathers start on day 8. And you can see the fine tail feathers develop around day 10 to 12. Now comes the time when the chicken is almost fully developed and the final stages of development take place, which include the withdrawal of the gut into the abdomen, the final formation of feathers, and the chick is now about to hatch. The chick uses the beak to break open the eggshell and then come out of the egg. So this movie gives you a complete picture of development of an organism from a single cell which divides into multiple cells. There is the formation of a blastula, gastrula, and shape and form in the form of a chicken is then comes about. Let's go to the next slide, please. Now very obviously chicken eggs are easy to get and fertilized eggs can be used to follow development. Development can be followed by taking the embryo out of the egg for about 2 to 3 days, not beyond that. But if in science class in your school, if it is allowed, you can open up chicken embryos at different stages and track development. Can we go to the next slide, please? Now what I am going to be doing now onwards is to talk about development, focusing on organism which I work on, which is shown on the left hand side. This is the fruit fly. It is called Drosophila melanogaster, and scientists like me usually choose organisms to work on which are easy to handle. For example, working with elephants, which is a picture on the right hand side, would be extremely difficult for many reasons, which include the fact that elephants produce very few children, they occupy a lot of space, eat a lot of food, and it will be very difficult to do development and genetics with elephants. So we use fruit flies. The picture you are seeing is a male fruit fly. The red parts are the eyes of the fruit fly, and in the next slide I will show you how we use the fruit fly. We grow them in small vials. The specks you see inside the vial are the developing larvae and pupae of the fruit fly. The larva, the vial is closed with a cotton plug which keeps the flies inside the vial, and at the bottom you see the fruit, the food which the fruit fly eats, which is usually a mixture of sugar and agar. Now in the next slide, what I want to convey to you is that when we study an organism as small as a fruit fly, it is about 1 millimeter in size. What I want you to appreciate is that you are effectively studying yourself, and the reason this is true is because, as I have already told you, all animals on earth are similar. We arose from common ancestors. The fruit fly, though small and humble, has eyes, has muscles, has a nervous system, it has appendages, and it has many behavioral traits which are very similar to humans. So when we study a fly, we are actually studying ourselves, be it be studying animal development or any other behavior in the fly. On the next slide, I will show you 2 ways by which flies are like us. The movie on the left hand side shows you that just like us, flies fight amongst themselves. Here are 2 males who are not happy with each other and engaging in a bout of warfare. They are upset about something. And on the right hand side, you will see a slow-mo picture of a fruit fly in flight. And what the right hand side picture, if you can click the movie on the right hand side, please. What the right hand side picture is showing you is how a fly uses its appendages, wings, to fly in the air, and obviously this flying uses the muscular system, the nervous system, and a lot of energy to stay airborne. Now in the next slide, in the next slide is the life cycle of the fruit fly, which is something I study. This picture is taken from the Manchester fly facility, and eggs of a fly take approximately 10 days to hatch into an adult, and in between they go through a larval and a pupal stage. Now, I would strongly encourage you to, for those of you who are interested in learning more about the fruit fly and its development, to go to the Manchester fly facility website and go to the YouTube channel. For teachers who are attending this lecture, the Manchester fly facility has very nice modules for biology for schools and Drosopher schools, which are really worth following and can be used in teaching. Now, rather than showing you a movie on the fruit fly development, in the next slide I will show you a movie on the life cycle of a monarch butterfly from the Chicago Nature Museum, which is very similar to the life cycle of a fruit fly. In this movie, you will see the egg go to a larva, then to a pupa, and then finally form an adult, which is a characteristic feature of developing organisms. Again, life starts with a small single cell, which is an egg which gets fertilized, and the cell starts to divide. At this stage, the egg, which has tens of thousands of cells, is formed into a larva, and the larva comes out of the egg, and it begins a life where it roams around on the plant. And at this point, in order to grow into an adult fly, adult monarch butterfly, it needs a lot of energy. So much of the time of the caterpillar is spent in eating leaves, so that it stores lots of reserves of energy for the pupa step and for the conversion of the pupa into an adult monarch butterfly. Here you see the young larval caterpillar chomping away on leaves, and it will grow in size, and it will go through multiple stages of molting, and with each molting stage the larva will become larger and larger. Once it attains the correct size and has stored lots of energy, it will enter into a pupal stage, which is a stage where it doesn't move, and the pupa will finally hatch into a beautiful butterfly. So here it's looking for a safe location to pupate, and here the pupation stage will start. The pupal case is now formed all along around the larva, and this is what the pupa will look like, and it will remain absolutely still for the next 1 to 2 weeks. Inside the pupa, wondrous things are happening. There is a change in shape and form, and what comes out of the pupa is a fully formed adult monarch butterfly. And to remind you all again, this is part of the process of development, animal development, which biologists follow, study and try and learn. For those of you who have questions, please do ask questions in the chat, and I will attempt to answer them. Meanwhile, development continues and we end up with a beautiful adult monarch butterfly, which will now live the final stages of its life cycle, reproduce and lay eggs, which will then go on to form the next generation of butterflies. And like this, butterflies will continue to remain on this earth. In the next slide, I will now move on to a few more advanced concepts in animal development. This is more for students who are in senior classes like 9th and 10th standard or beyond. Now, there are three things I want to leave you with before I end this seminar. The first is that foundations are very important for development, and a major foundation for successful animal development is the formation of the body axis, and this is something I will explain in the next few slides. The second thing is, can I go back one slide? The second thing is that we have a large number of genes in our genome. For humans, it's about 25 to 30,000 genes. For insects, it's approximately around 25,000 genes, and these genes and their function is very important for development. And one way of studying what each gene does in our body is to make mutations in these genes, which means that we destroy these genes and study how the absence of these genes affect development. The third thing which I want to leave you with is that the mechanisms which are there in development, the processes by which an animal develops, are very, very similar, whether we are looking at invertebrates or vertebrates. And by looking at one kind of animal, be it a frog, be it a fish, be it chicken, we can understand a lot about human development, for example. So, let us go to the first part. Now, I will give you an analogy over here. Here is a picture of the world's 10 tallest buildings. These are really very tall buildings, over 800 meters in height, and in order for these buildings to be to not fall down, they need to be built extremely well with the modern engineering concepts, and they need to have a strong foundation. In the next slide, you see an obvious example of what happens when a building is designed with either designed badly or has a weak foundation. You can see that this building has fallen down. Now, just like engineering a building and engineering or designing a building, in animal development, which you see in the next slide, the foundations are very important, and these foundations are laid in the very early stages of development. And one of the main themes of the foundation in the early development is to determine the body axis. Now, what do I mean by the body axis? You, me, all animals, multicellular animals, have an anterior side, a posterior side. So, this is my anterior, my posterior is near my legs, I have a front which is called the ventral side, and I have a back which is the dorsal side. Now, the establishment of these axes turns out has to happen very early in development, and every cell in the body orients itself and determines what it is going to become based on its position in respect to these coordinate axes. I will call them the anterior posterior or the AP, and the dorsal ventral or the DV axis. You can see the axis is drawn for a horse and also for a fruit fly. Now, if you notice, the fruit fly has a very specific pattern. It has a head on the anterior side, it has wings which are on the dorsal side, it has the fruit fly is segmented, it has T1, T2, T3 which are called as thoracic segments, it has an abdomen which is labeled A1 to A8, it has legs and it also has wings, and all these structures, including the nervous system and the musculature inside, are developed based on their position with respect to the axis which I have drawn. In the next slide, let us see what is an example of what happens when the axes are not positioned properly. Here is a picture of a larva or a tadpole developing from a frog egg, and as you can see, something has gone wrong over here. The anterior posterior axis is split, it is confused, and instead of forming a single head, this developing organism has formed two heads. And similar problems in shape and feature can happen when development does not proceed normally. So, the formation of the axis is very, very important. The next slide. Now, again to re-emphasize, may you be studying human biology, or you may be studying Drosophila developmental biology and or any organism in between. Axes are very important. The axis, the DV and the AP are set up very early in development, and in the absence of this axis, development cannot proceed. Any minor modification to the setting of this axis will lead to a stalling of development. The next slide, please. Now, let us go to the first point, which is the setting up the foundations or the axis in development. What you see over here on the top is an egg or a developing egg, which in the abdomen of a female fly. The cells on the left hand side with blue markings on them are called the nurse cells, and these are the mother cells. And what these mother cells do is they deposit maternal material. They deposit food into the egg, so that the egg can prepare for its future after it fertilizes with the sperm. In the pictures below, you can see that the AP axis on the left hand side is set up in the egg, which is a single cell, and the DV axis is set up on the right hand side. Now, what I will try and convey to you is that RNA and protein is expressed inside these embryos, and these proteins are not distributed evenly. They form gradients, and one such gradient is of a protein called bicoid on the left hand side, and another gradient on the right hand side in red from the bottom to the top is of a gradient of the protein called dorsal. Now, these protein gradients are what which end up specifying the coordinate axis in animals, the AP and the DV axis, and all development proceeds after these foundational axes are laid. Can we go to the next slide, please? Here I will give you an idea of the concept of how these gradients are set up. For example, the bicoid protein is set up on the left hand anterior corner of this large egg, which is shown in front of you. The protein is made on the extreme left hand side, and it diffuses from the left to the right hand side. To remind you of what diffusion is, all you have to do is to take a little bit of ink and put a drop of ink in a glass of clear water, and you can see that the ink particles will diffuse. In a similar way, the bicoid protein diffuses from where it is produced on the left hand corner of the embryo, which is the anterior side, and it diffuses from the left to the right hand side. And similar proteins are made at different parts of the embryo, and they diffuse from the area of production, and the concentration is highest near the area of production and lower as you go away from the area of production. In the next slide, you will see a picture of this diffusion happening of a protein in a live embryo. On the left is the first stage, second marked in 2 is the second stage, and the third is the third stage. As the stages go from 1 to 3, the protein diffuses from the left hand side towards the right hand side, with highest concentration of protein on the left hand side and lower concentrations of protein as you go away from the left hand anterior part of the embryo. In the next slide, what you will see is how these gradients are used to separate what cells are going to become. So, if you imagine that initially, as shown on the top, there are about 8 cells shown, which are going from the left to right. Now, the moment there is a gradient of a protein formed with highest on the left hand side and becoming less and less from the left to the right, each cell which is present in this embryo will receive concentrations of different states, high concentration of bicoid, low concentration of bicoid, or no bicoid, and similar gradients will be present from the right to left and from the top to bottom. Based on these different concentrations, each cell which is there inside this embryo will decide that it is going to become something completely different. One cell will along with its neighbors will become a heart, another group of cells will become neurons, another group of cells will form the leg, and all these decisions are taken completely based on its position in the coordinate axis, which is the AP axis or the DV axis, and these coordinate axes are defined by protein gradients. The next slide shows you how these protein gradients define thresholds in the embryo. So, at a different concentration of protein gradient, for example, defined by threshold 1, all cells which are marked in blue will form the head, all cells marked in, let us say, white will form the thorax, and all cell marks in red will, let us say, mark the abdomen. So, this is how along the anterior posterior axis, depending on multiple gradients and the thresholds which are kept at different concentrations of gradients, animal makes the head, the thorax and the abdomen, and the animal decides whether it is going to make the back part, whether it is going to make a nervous system, or whether it is going to make the skeleton in an animal. In the next slide, you see a picture of all the different major gradients in the fruit fly and the early embryo. These are the bicoid, the hunchback, the caudal and the nanos gradients. If we go to the next slide, I will show you a movie of early development immediately after these gradients are formed. So, the gradients are now just completed, the cells are now going to go into gastrulation. It is a very dynamic state in animal development, and at the end of these gastrulation, the head and the tail and all other structures will be formed, and the animal becomes a segmented animal. Now, as you can see on the top part of the embryo, somewhere near the bottom where the arrow is pointing, what you will see developing, and it will happen in the next few seconds, is the nervous system of the animal. For invertebrates, which the fruit fly is, the nervous system is on the front, which is the ventral side of the animal. In vertebrates such as you and me, the nervous system is formed on the dorsal or the back side. The twitching you see in the animal right now is the nervous system contacting with the musculature of the animal, and the animal is now learning how to move, and in another hour or so the animal will hatch out and it will become a larva. Let us go to the next slide. What you could not see in this movie is an interesting feature which you will study in 8th standard, I believe, which is the blastula, which is where we started the movie, based on mechanical forces at the bottom, showing folding of the cells into a invagination, which finally leads the single cell blastula on the left becoming a multi-layered gastrula on the right, and in combination with all the movements which you saw in the movie just before, the first steps towards forming an animal would have taken place. If you go to the next slide. Now, this is how, with all these proteins expressed, that the anterior posterior axis of an animal is formed and the dorsoventral animal of the animal is formed. And I must emphasize and underscore this is very important, that it is the formation of these coordinate axis, the AP axis and the DV axis, which is very, very important. It lays the foundation for all movements of cells, and it lays the foundation for every cell to decide and define what it is going to be in the adult animal. Let me go to the next slide. Now, I am going to end with two concepts. The first is that genes are very important for development, and all the movies you saw, all the movement of cells, the formation of the nervous system, all these stages depend on genes which are active and are defining how these events occur. Now, in 1995, a Nobel Prize was given to these two gentlemen and the lady for doing a lot of very interesting research on how genes are important for defining development and important for every stage in development. In the next slide, I will show you a movie of some work done in my lab, and on the top movie is a normal embryo showing development, and it will go through the stages, and everything you see, this is done with a normal microscope, is fairly normal. But in the bottom you see a mutant, if you can go back, and the bottom movie, if you could play again, is a mutant embryo where the normal development cannot proceed and all kinds of chaos happens in this embryo. This is a mutation in a single gene, and development stalls much before larval formation takes place. So, there are many such mutations known, which if a gene is destroyed, the animal cannot develop further. And in the next slide, it is not only in early development, but also in late development. Here are two examples. On the left is a mutation in a gene called IIS, and if that gene is destroyed, you can see that the fruit fly on the left does not have an eye at all, no eye is developed, it is completely blind, but it can go through its life cycle. And on the right hand side is a even more stranger mutation. Here the antenna, which is normally present on the head, is replaced by a leg, and it is called the antennapedia mutation. And these kind of strange changes of shape and form can happen when an important gene does not function normally, and in this case the antennapedia gene does not function, and instead of an antenna, a leg is formed because the animal gets confused. So again, to emphasize, genes are responsible for development, and we can study what each gene does for development by making mutations. Can you go to the next slide, please? Here is another example of how important genes are. Here we have done a genetic engineering experiment. The gene which is responsible for formation of the eye is misexpressed. What I mean by that is, we forcefully express this gene in different body parts. This is not done in normal development, but scientists can do this kind of experiments. And when we force expression of the genes all over the body, you can see that eyes are forming all over the body of the animal. This is not normal at all, but this shows you how powerful genes are and how the expression of a gene can target any tissue to take a development fate, in this case the formation of the eye where it is not supposed to be forming. In the next and final slide, I will end by telling you that all life forms are similar. This is what I started with. I have given you an introduction to animal development through a series of movies and images, and I hope by looking at this slide you appreciate the fact that may it be dogs or humans, may it be fruit flies, may it be cockroaches, may it be an elephant, the developmental mechanisms are very similar, the process of development are also very similar. We all start life from a single fertilized egg, which we call the zygote. This zygote divides into many cells. The number of cells may vary between different organisms. They can be in the few thousands to a few million. As the cells divide, they go through processes which involve processes like gastrulation, and these groups of cells take shape and form into different organisms, ranging all the way from seahorses, dogs or humans. The number of cells in each organism is very different, our shape and form are different, but finally we are all related, and the developmental mechanisms, the process of development is conserved between all life forms. So I will stop here, and in the next slide show you resources where you can go and see many interesting features and ideas about developmental biology. Please just write down these words like Society for Developmental Biology or the Manchester Fly Facility or the DNA Learning Center into your Google bar, and you will be taken to very nice educational sites. This is true for students who are listening to this seminar as also for teachers. These educational resources are very simple. They are movies or very simply explained concepts in developmental biology. All these sites have been made by working scientists, so they are very accurate and very up to date, and I hope that my seminar would spark an interest in developmental biology in all of you, and will lead you to further explore the very beautiful world of developmental biology. I will end by thanking all the people who are responsible for setting up this seminar in the next slide. Again, to remind you that I am coming live from the Science Activity Center in IISER Pune, from the Center for Excellence in Science and Mathematics Education. I will end by answering a few questions which have been asked on in the text boxes. So, one of the questions from Manisha Bhandari, I am moving to questions now, is how do the cells get energy to move inside? That is a very important question. If I will give you one example, if you look at the development of the frog embryo, The frog embryo is laid by the female frog, and part of the process of getting the embryo ready for further development is the female deposits a lot of yolk protein inside the embryo. This yolk protein is used as an energy source and is used up during the development of the animal. Remember that frog egg develops outside the body of the animal. So, the mother frog deposits a lot of energy rich proteins and carbohydrates, which will allow the animal to go through all stages of development and hatch into a tadpole. Once the tadpole is formed, the tadpole can eat and gain energy for itself. Another question is about how and when did life start on this planet. This question is answered in the first few slides. It is believed that life originated on our planet approximately 3.5 to 4 billion years ago, and the initial forms of life were very simple, but we have now evolved into very complex life forms which are present on this planet today. One of the questions asked is, what happens if we remove the outer lining of the pupa during development? Well, this will not allow development to continue. The reason is the pupal case shields the animal which is going through a very transformative stage of development from the outside world, and the outside world includes microorganisms, viruses, which will interfere with the process of development. So, think of the pupal case as a shield inside which the animal is growing, and it is very important that if you want the animal to grow successfully, that you do not disturb animals which are growing in nature. Another question is from Sanskruti, who asks, what is the lifespan of a butterfly? I do not know the exact answer. I think you can look this up on the web, but fruit flies, which is something I am comfortable with, which is also an insect, can live as an adult animal up to 90 days. So, butterflies will also have lifespans around the period of 1 to 2 months as an adult animal. One question from Dipali is about whether what comes first, the egg or the chicken. Now, this is a philosophical question. We really cannot answer this question. We know that there is a life cycle. In the life cycle, there is an egg and there is an adult. The adult makes the egg and the egg makes an adult. This is the way life cycles have evolved over the last million years, and this is a question which we do not answer as developmental biologists. What we do understand is the process of how the egg makes the chicken. So, these are all the questions I have as of now, and I will stop here. Thank you very much for attending this class. Thank you.